This work is not glamorous, but it could shape the next chapter of how we could approach food production. I'm a veterinarian specialized in food microbiology. I deal with bacteria in general that can cause disease in different animals and humans. Salmonella infantis is one kind of bacteria that can not be dangerous in animals, like for example poultry, but that can be dangerous to humans through the food chain. These are Salmonella infantis taken from chicken gut. Salmonella can cause infections, diseases like abdominal pains and diarrhea, and fever sometimes. The industry generally tries to keep bacteria at bay through the use of antibiotics. Antibiotics have been used for decades to kill bacteria or to stop them growing. But once bacteria are exposed again and again to antibiotic therapies, they can get resistant, and that's one of the reasons that day after day, antibiotic after antibiotic, bacteria are getting more and more resistant to them. If antibiotics stop working, we can face serious problems in fighting bacterial disease in humans. In this buffalo poo is a potential solution. Microorganisms able to attack bacteria and destroy them. What we are hunting today are so-called bacteriophages that comes from Greek. Bacterion means bacteria, small stick, and phagos means to eat. So we are looking for microorganisms that eat bacteria eaters. Animal wastes are a very good place to search for phages because that waste is full of bacteria, and wherever bacteria exists, bacteriophages exist too. I think that this is a weapon that nature put in place to contrast the overgrowth of bacteria in the world. Antibiotics can be a blunt tool because they stop all growing bacteria, even the good ones. Bacteriophages are very specific, like a key in a lock. They kill only specific bacteria. It attaches on it and injects its material into the cell and hijack the cell to reproduce other bacteriophages and destroy the cell. This morning we were in the field. We took some samples of feces, and in the lab we need to separate bacteriophages from the rest of the sample. And then we prepared some plates in order to check the effects of bacteriophages.
So, Giuseppe, I just prepared the chicken skin, and what I'm going to do is apply 10 microliter of the known Salmonella. Giuseppe is preparing the phages, isolating them, and we test how we can apply the phages in real life. My name is Georg Duscher. I'm from AGES, the Austrian Agency for Health and Food Safety. Through the European Partnership of Animal Health and Welfare, this work moves back and forth between us. We placed Salmonella in a defined dose on the chicken skin. We choose chicken skin because it has a lot of pores and ridges where bacteria can hide. So we wanted to see if bacteriophages are able to enter these pores and kill the bacteria. We had one group which we treated with bacteriophages, the other we left untreated. We incubated the chicken skin for 24 hours. Thereafter, we took a swab. What you can see now is really that the Salmonella group did grow, and we hardly can see any Salmonella on the phage plates. The treated group had a reduction of bacteria. Phages were able to reduce the bacteria on the chicken skin. It's good, very good. Aerosolic application of bacteriophages to really broaden the spectrum of the use and the treatment. You can use it in stables, animal facilities, or even use it for inhalation treatment of patients. We are just at the beginning of this study, so we need to be really careful. But still, this experiment showed us some promising results. The phages are a potential pillar in a broader strategy to fight antimicrobial resistance.
So in the sample, bacteriophages were present, and they act just like antibiotics in this particular disc. Thanks to the European Partnership on Animal Health and Welfare, we can work together to improve animal health. Bacteriophages are not meant to replace antibiotics, but they can be a complementary tool to preserve the use of antibiotics in the future.